Hello all. Now we are discussing about the factors affecting fermentation. Fermentation. Fermentation is a metabolic process that produces chemical changes in organic substrate through the action of enzyme using microorganism. Fermentation can be affected by two types. First one is a physical factor, temperature, pH, osmotic pressure, hydrostatic pressure and radiation. Second one is the chemical factors, oxygen, carbon, nitrogen, phosphorus, sulfate, etc. are there. First is the physical factors, temperature. Temperature is the most important factor that determine the rate of growth, multiplication, survival and death of all living organisms. High temperature may be reduce fermentation process because of damage microbes by denaturing enzyme, transport carries and other proteins. Microbial membrane are disturbed by temperature extreme. At very low temperature fermentation process may also reduce because membrane of bacteria may also solidify and enzyme also do not function properly. pH refer to negative logarithm of hydrogen ion concentration. Fermentation process may also be affected by pH because of microbial growth is strongly affected by the pH of the medium. If a drastic variation in cytoplasmic pH occurs, then it may disturb the plasma membrane or inhibit the activity of enzyme and membrane transport the protein. Next is a osmotic pressure. Osmotic pressure is a minimum pressure which needs to be applied to the solution of the prevent inert flow of water across a SPM. Solution may be hypotonic, isotonic or hypertonic. Water activity of solution. Water activity equal to vapor pressure of solution by vapor pressure of pure water. Classification of bacteria according to osmotic pressure. First one is osmotolerant, are those microorganisms which can be grow at relatively high salt concentration. Example, Aromonas species, Staphylococcus, etc. Second one is allophilus, growth in presence of salt concentration above the 0.2 to 0.6. Example, Halobacterium, Halobium. Next one is hydrostatic pressure. This is the force acting per unit area. Classification of bacteria on the basis of hydrostatic pressure or as barophilus bacteria which grow at moderately high hydrostatic pressure. Example is Halomonas celeria, Xenoporus, etc. Barotolerant does not get affected by increased pressure. Next is the radiation. Sunlight is the major source of heat. Infrared is the major source of earth heat. Ionizing rays can produce mutation which may result in death. Visible light is beneficial because it is the source of the energy of photosynthesis. Next is the chemical factors. First one is a carbon. Simple to complex carbohydrate can be added to media in fermentation as a source of carbon. Different sugar like mannitol, sorbitol, organic acid, fatty acid, protein, peptides can be added. The selection of carbon source depends upon the availability as well as cost of material. In most of fermentation media, crude source of carbon is added. Example of simple carbohydrate are blackstrap, molasses, corn molasses, beet molasses, sulfide waste liquor, etc. Carbohydrates are starch, corn, rice, potatoes, etc. Next is the nitrogen source. Nitrogen sources are required for most organelles to synthesize protein, nucleic acid, and other cellular components. Nitrogen may be provided as bulk protein, such as soya meal, as pre-digest polypeptides such as peptone or tryptone or ammonia or nitrate salt. Salt of area, ammonia or nitrate can be used to nitrogen source. When fermentation organisms are not proteolytic in nature, pure form of urea, ammonia and nitrate are used as nitrogen source. When fermentation organisms are proteolytic in nature, animal and plant raw material used like casein, cereal, grains, peptones, etc. Next is oxygen. Depending upon the amount of oxygen required by the organism, it may be supplied in form of the air containing about 25% volume by volume oxygen, or occasionally as pure oxygen required are particularly high. The organism oxygen required in may vary widely depending upon the carbon source. Most of the fermentation process filters sterilized air is supplied into bioreactor. Next is a chemical factors on the water. All fermentation process except the solid state fermentation require vast quantity of water. Water is not only major component of all media, but it is important ancillary service like heating, cooling, cleaning and rinsing. A reliable source of large quantity of clean water consist composition is therefore essential. Important factor of considers when assessing sustainability of water supply are pH, dissolved solid and effluent contamination. Thank you for watching our video.